and together we will be looking at a few exam questions and explaining some of the important concepts regarding to these questions that you might experience in past papers. So let's dig into it.
Right, so the first thing we need to do is just quickly recap the structure of the eye. So right on the outside, the cornea at the front part is responsible for the greatest amount of refraction of light rays. Remember that as a receptor organ, your eye wants to converge light rays on the retina in order to observe or see what you're actually looking at. And the cornea is responsible for that first initial breaking of light, and it's a completely transparent structure that cannot change its shape. Then we have the suspensory ligament there, which is attached to the lens and actually holds the lens in place, helping to change the shape of the lens during the process of eye accommodation. The pupil is an important component, not that it's an actual structure, but rather the absence of a structure. It's that opening in the middle of the iris which changes shape and size to allow more or less light to enter the eye. And the pupil mechanism is a very important part of this chapter that's often asked in exams. Then the aqueous humor found in our anterior chamber is responsible for nourishing the lens and the cornea, which have not got their own blood vessels to supply them with oxygen and nutrients. And it helps to also keep this area moist, supporting the cornea in the front part of the eye. Then the iris is the colored part of your eye, whether you have green or blue or brown eyes. The pigment in the iris determines that. And this is the part that controls the size of the pupil with the two muscles that it contains, which Yolandi will quickly recap with you. Then the lens is a very important structure, essential for what is known as eye accommodation, focusing light rays on the retina. The lens is called biconvex because it has two sides that are bent and that refracts light or bends it. And it's also an elastic structure. So unlike the cornea, this can actually change its shape in order to adjust how much refraction occurs. It's also transparent, so it doesn't have its own blood vessels to supply it with nutrients, hence the aqueous humor. And this is a good thing because the lens wants to allow light to pass through. Then at the back, we have the vitreous humor, kind of more like a jelly-like substance, helping to keep the shape of the eye, maintaining pressure, and making sure that the sclera, the choroid, and the retina stay attached to the back of the eyeball. Then the blind spot is the area where our optic nerve enters and leaves the eye, as well as many blood vessels supplying the choroid with blood. The blind spot is an area where no vision can be interpreted because there are no rods and no cones, no photoreceptors to pick up on light. Then the yellow spot is the opposite of the blind spot. This is actually the spot where you want light rays to focus because it contains the greatest number of cones, the photoreceptors responsible for giving you sharp, bright, clear color vision. So that's where images form the clearest, sharpest image. Then the choroid is the dark part of the eye at the back, which is filled with blood vessels and also a very dark pigment, which helps to prevent reflection of light rays in the eye. And then the sclera, the white outer part of the eye that we can all see when we look in the mirror, a connective tissue layer that helps to protect the eye against damage and keep it in its shape. And then last but not least, the very important retina, which contains the two photoreceptors, the rods and the cones. The rods responsible for dim light conditions, recognizing black and white vision, and the cones being active in bright light conditions, allowing us to have color vision, clear, sharp images.
Okay, I will be running through the two mechanisms associated with the eye. The first one that we have is pupillary mechanism. So in this case, it is where we are changing the size of the pupil based on the amount of light that is present in the environment around us. So I'm sure that you guys would have seen that if you go into bright light, the size of your pupil becomes smaller, as opposed to when you walk into a dark room, your pupil will become larger. Now remember, the pupil itself is not a structure. It is literally just the hole that allows light to come in. So in order to change the size of that hole, we need the iris, which is the color component of the eye, to manipulate through two sets of muscles, being radial muscles radial muscles and circular muscles, and they will be antagonistic to each other. So in order for the one to work, the one needs to relax and the other one needs to contract, hence being antagonistic. So we're going to start off with our scenario. We're going to run through the steps surrounding it. We have a situation where there's bright light. We can see in the image here that the size of the black hole, the image, the pupil there, it is very small. And the reason being we want to limit the amount of light that enters through the pupil to our photoreceptors in the back chamber so that we don't damage them, because the excessive amount of light can actually destroy our receptors. So in order to reduce the size of a pupil, we're going to have our antagonistic muscles working together. Our radial muscles would need to relax. Our circular muscles would need to contract. So that's easy. Our radial muscles and alpha relax. Circular muscles would need to then contract. CC here yet again. So these two guys will obviously then allow for that diameter of that pupil to constrict. So we use the word pupil constricts. You can use the words it becomes smaller, but you might just end up getting an extra bonus mark for saying that the pupil constricts. And this will therefore result in less light entering your eye. So quite often they ask this question in the exam, and everyone talks about how the radial and the circular muscles work together and the pupil becoming smaller. But the cherry on the cake is kind of like the less light will enter your eye, which is the aim of the pupillary mechanism. Now, in the event where we have a dim light situation, so you walk into a dark room, we find that our pupil will dilate, which means that it enlarges. The reason for it, we want as much light to enter through into our back chamber so that the photoreceptors can be stimulated and we can actually see as much as possible in dark environments. So in order to achieve this, we're going to have our radial muscles of the iris will contract and our circular muscles of the iris will relax. So that is exactly the opposite to what we've seen in a light environment. The consequence of this antagonistic action is that the pupil will dilate and more light will enter the eye. Now, when you are explaining this situation, throw in the words that these two muscles, the radial and the circular muscles, work antagonistically to each other. You might just end up getting another bonus mark for using a fancy word in your exams. Right, and then being able to draw these images as we see it in the two different environments, there might end up being another exam question about that as well. So indicating that the pupil has enlarged, it is dilated within the dim light situation. Right, the next one that we have is accommodation. And the process of accommodation is all about how the lens has to change shape when someone is coming towards you. So we need to be able to remain focused as objects are moving towards or away from us. So what will happen is that the lens itself will either distend or will become rounder, for instance, or will become elongated based on the movement of the object. So whenever something is moving 6 meters and closer, the process of accommodation really kicks in. And that is where your ciliary muscle needs to work, contract in order to change the shape of the lens. And the same applies. If something moves away, the ciliary muscle will relax, and that will also bring about the change within the lens. So I always think about accommodation. The word accommodation means someone is going to a house and they're staying over, and therefore I make that association with movement. And that makes it easier for me when I get an exam question. Is the exam question asking me about the pupil size, pupillary mechanism, or is it telling me something about something is moving towards me or away from me? And then that will be the accommodation component to it. So they will ask you, are we working with pupillary mechanism? Are we working with accommodation in a question? So if we run through the process of accommodation, if we go to our presentation quickly, we can see, if we start off, accommodation is a series of changes that takes place in the shape of the lens and the eyeball in response to distance of an object from the eye. So that is the definition of it. We're going to start with distant vision quickly. So this is now objects that are further than 6 meters away from us. Now, anything that is far away, it is actually less work for your eye to look at something which is distant. So we find that our ciliary muscle is relaxed. Now, this is going to be found right here inside of your ciliary body. So the body is literally just like the whole bulged area that's attached to our suspensory ligament, but it's the muscle inside of the ciliary body that brings about a change. So we find that the ciliary muscle, when it's relaxed, means that the ciliary body moves further away from the lens. So I think about a situation where someone's making a bicep, the bicep bulges outwards, but if the person doesn't make a bicep, contracts the muscle, the bicep kind of like flattens out. Now, the same thing is happening inside our ciliary body as well. Whenever it contracts, it makes a bulge. Whenever it relaxes, it flattens out. Now, because these suspensory ligaments are attached to that body, when our ciliary body flattens out, it means that our suspensory ligaments are going to be stretched out. They become tight. We can say they become taut. And that obviously puts tension on our lens. So it has like a knock-on effect. So the tension of the lens increases, it's being stretched out more. We said that the lens becomes less convex in shape. So it's important that we use this descriptive word when explaining what happened to the lens. This will result in the light rays are refracted less. And to end it all off, the light rays are focused on the retina and the image falls exactly onto the retina. So as the light enters, it's going to bend, and we want it to fall exactly on the retina at the back of the lens. So that refraction of light is going to take place, and obviously clear vision will be formed. Now, when we're working with near vision, we are talking about anything that is 6 meters and closer. So if you spend the whole day reading a book, or if you're on a computer for the whole day, your eyes are actually taking strain. Meaning, and I'm sure that you've heard about your eyes, or someone said that your eyes are strained. It's all about the fact that your ciliary muscle is in a constant state of contraction. So if you've spent hours and hours and hours reading your book, you actually need to rest your eyes a bit, because this muscle is working very hard. So when that muscle is contracting, the ciliary muscle, it bulges outwards, as we've mentioned before, and as a result, the ciliary body bulges outwards. It means that it's moving closer towards the lens. Now, when it moves closer, the suspensory ligaments will become slack. The consequence is that the tension in our lens will decrease. The lens will become more convex in shape, which means that the light rays, that when it enters, it bends more, and it's going to fall onto that retina. So the light rays will be focused onto the retina, and a clear image will be formed. And that's the process of accommodation.
Let's look at an example of an exam question where they give you a picture of the human eye, and the first thing they want you to do is to give a function of the different parts A and C. So first look at the diagram, and let's try to identify what we have been given. So we can see that there are 3 labels. Iris has been provided. B shows me the gap in between the 2 sections of the iris, so that's the pupil. A then represents the lens, and C is indicating the retina. So one function of the lens is to refract light in order to focus those light rays on the retina. That is its main responsibility, and that's what it does during eye accommodation. C, your retina, is the place that contains those photoreceptors, but that's not a function, that's a structure. So you must basically give the function of the photoreceptors, which is to receive light stimuli and to convert them into impulses. Then they ask a quite nice question. They're telling you that nocturnal animals sleep during the day and are active at night, if you didn't know what nocturnal animals are. And they want you now to think about animals that are more active in darker conditions and explain how part B for such an animal will differ. Now, part B being the pupil in this diagram, for a nocturnal animal that's more active in dim light conditions, like Yolandi explained, you want to have as much light entering the eye in those circumstances, so you will actually have a pupil that can dilate even more to allow more light to enter the eye. And that's why you can actually see better at night. The second part then asks about C, referring here to the retina, if you look at the picture there. They're asking you, so in a nocturnal animal, what would be different with regards to the retina of the eye? Now, this relates to the functioning of the photoreceptors, so the rods and the cones. Cones, I've mentioned, are responsible for bright light conditions, looking for color vision. Rods are active in dim light conditions, allowing you to see black and white. So you can imagine that in a nocturnal animal, color vision is not that important for them because they're going to be moving around in situations where there's not a lot of light. So in their retina, it would be amazing adaptation for them to have much more of the rod photoreceptor so that they can actually see better in darker, dim light conditions. And that's where you also get your mark for this question. So you see that for C, the answer then is that the retina has to have more rods which are active in dim light conditions so that they can see in those conditions. And then that typical 4-mark question that Yolandi has gone through to describe how the iris controls the amount of light entering the eye when a person is exposed to bright light. So for bright light, I always like to think of a room. You're sitting in a room and there's too much light coming in and you close the shades, right? You want less light to enter, so you close the opening between the windowsill and the shade, so the pupil has to do this. It has to constrict. And if you do this with your hand, you're making a circle. So circular muscles have to contract and radial muscles have to relax. So let's look at the answer here. So just like Yolandi explained it, radial muscles of the iris relax, circular muscles of the iris contract, the pupil becomes smaller, so you constrict it, and then less light will enter the eye. Another classic example is where they give you a section through the eye like this, showing you the lens being in different states. So they're showing you in diagram 1 a lens that seems a little bit flatter. You can see that the suspensory ligaments are more tight compared to diagram B, which has a more rounder lens. So this is definitely looking at eye accommodation. So give the letter and the name of the part that contracts to change the shape of the lens. So there are only 3 basic muscles in the eye, the ciliary muscle and the 2 in the iris. So if this is accommodation, the one that's going to contract will be the ciliary muscle indicated by A on our diagram. This name and the letter of the part that controls the amount of light that enters the eye will be the iris, C, because it's not about the pupil that's actually controlling how much light enters. That's just the hole. Remember that the iris is the muscle that changes the size of that pupil. And then lastly, that is protected by the conjunctiva, that delicate membrane over the surface of the eye filled with pain receptors. So that has to be the cornea, indicated by D, our very outer layer. Then they continue by asking you to study the 2 diagrams and ask you what process is responsible for the change in the shape of part E. So part E, you know, is the lens in this diagram, and we've discussed the fact that the change that it undergoes is referred to as accommodation. Then they ask you which diagram, 1 or 2, represents the state of an eye when a person is reading a book. Now, a magnifying glass is a round object, right? And it's something that you use when you want to look at a close object. You want to study ants. So I like to remember that the rounder the lens, the closer the object is to the eye, like a magnifying glass. So if you look at the 2 diagrams on the slide again, you're going to see that diagram 2 there has the rounder lens. So the flatter, not the flatter lens like diagram 1, which is essential for farther vision.
Okay, in this question we have one which is yet again linked to the diameter of our pupil, so you have to make that immediate connection with pupillary mechanism. And they even tell you in the description of the question. So the graph below indicates a change in the diameter of the pupil of the human eye. So over time, we found that the diameter went from being quite large, about 6 millimeters, and then suddenly something happens where it reduced in size. So we know that this something that is happening here is where a person has gone from a dark room where the pupil is max like size diameter-wise to being very constricted. So definitely the person has experienced bright light here. And then it goes back to being slightly more dilated, indicating that maybe it's become a bit cloudy or has gone to an area where, there's a little bit more shade. So for question 7.1, they want to know which structures in the human eye is responsible for the changes, and that would be our iris. Question 7.2, during which period of time was the person moving from dim light to bright light? So this will be the dim light, and it's moving into bright light. So then our answer, if we read it off of our x-axis, that will be between 20 and 30 seconds. And then obviously include the unit of measurement as well. Right, the last two things that we quickly want to run through is our eye defects, which is short-sightedness and far-sightedness. So when a person is considered to be short-sighted, we said that the person can see nearby objects but cannot see distant objects clearly. And here it's important that we don't finish our sentence with the person cannot see distant objects full stop, because that will mean that distant objects, the person is blind, which would be weird. Okay, that doesn't happen. It's just very blurry, and it's got ranges, degrees of being blurry. So we have to finish off the sentence by saying distant objects are not as clear. Right, so if we go to the process, this is representing a normal eye. So what we want to happen is the light rays should enter the eye, should refract, and it should fall onto the retina at the back. That's normal conditions. But in the state of where the person's got a myopic eye, short-sightedness, myopia, then we find that the light rays kind of falls short of the retina, so in the middle of the chamber, and the photoreceptors are not stimulated enough to give us a clear image. So the causes could be that the eyeball is either too long, or the cornea is too curved, or the lens is unable to become less convex. So we've got a solution for that. We give the person a concave lens, and that will refract the light a little bit more before it actually reaches the eye and the lens itself. And that will basically ensure that the light rays are refracted to such an extent that it falls on the retina in the back. Now remember that in grade 12 they can ask you what the corrective lens would be that you're going to use. I make the association when it's a concave lens. If I'm in the cave, I can only see what is right in front of me. So concave for short-sightedness. In the case where we are looking at far-sightedness, we can see distant objects clearly, but we cannot see nearby objects that clearly. So this is now the opposite situation. So under normal conditions, yet again, we want that light ray to fall on the retina, but in hypermetropia, as the condition is called, the light rays are refracted beyond the retina. So yes, photoreceptor stimulation will take place, but it won't be perfect, and the light ray focal point is behind our retina. The causes could be that the eyeball is too short. So in the case, if the eyeball had to be extended to this point, it would have been fine, but it's not. The cornea is not curved enough, or the lens is unable to become more convex. And in order for us to fix this situation, we provide the person with a convex lens. It will refract the light before it reaches the eye, and as it moves through the lens, it will perfectly form and accumulate on the retina at the back, ensuring that a perfect image is created.
So once again, an example of an exam question regarding the human eye and a visual defect. So they show you the diagram of how the light is refracted in this person's eye. And what we notice is that the light rays are focusing behind the retina. So this means that not enough refraction is happening. They're not being bent enough. It's taking too long to find the focal point. So this is a lens that is too flat. This is typical of someone with far-sighted or long-sighted vision. Now, once more, they give us indications of letters, having to identify them. B on the diagram is the lens, and then C on the diagram is indicating that dark pigmented layer in between the retina and the sclera, the choroid. Now, I've already answered question 8.1, which asks you to name the visual defect illustrated. So please go and look at those diagrams of eye defects so that you can recognize them. But far-sighted vision or long-sightedness is where there's not enough refraction. The light rays are going a long way before they find their focal point. So they're focusing behind the retina. The consequence of this, as Yolandi has explained, is that important difference between they cannot see the far object clearly. The image is blurred. So it's not that they can't see the object at all. They're not blind, but the image is blurry and they can't see it clearly. Okay. Explain the consequence if the light rays were to fall on part A. Now, part A in the diagram indicates the blind spot. It's where the optic nerve enters and leaves the eye. So when light rays fall there, no vision or image can be interpreted because there are no photoreceptors. So for 3 marks, you would have to say that there are no photoreceptors, so no stimuli would be able to be observed, converted into an image or impulse, and therefore your brain would not form any image in that area. Then they ask you 2 ways in which our lens is structurally suited to perform its function, and you must explain these 2 ways. So it means if you know the structure of the lens, tell me its best characteristics and what they are important for. So a few of the things I've mentioned right at the beginning is that the lens is an elastic structure. So that means it can change shape and therefore it can refract light. It can allow for accommodation. Secondly, it's a transparent structure. It has no blood vessels, which is excellent for allowing light to pass through onto the retina. And lastly, it is a biconvex structure, which means it is a bent surface that allows for the refraction of light. And then, as Yolandi explained, what type of lens would you prescribe for someone with far-sighted conditions? If the image is formed behind the retina because of too little refraction happening, we're going to need a lens that will refract even more, and that is a convex lens. Moving on to the ear then, we're quickly going to run through the structures that are important. So basically your ear is divided into 3 sections. The outer ear, running from the pinna up to the tympanic membrane, is an air-filled area. Then we have the middle ear, running from the tympanic membrane up to the oval window, including the eustachian tube at the bottom, and that's filled with air. And then the inner ear, consisting of the cochlea and the vestibular apparatus and nerves running to and from the brain. And this area is filled with fluid. So the pinna concentrates the sound waves down the external auditory canal. The tympanic membrane will vibrate to cause those little bones called the ossicles to carry those vibrations onto our oval window. The oval window will cause waves in the endolymph and perilymph of our vestibular apparatus and cochlea, leading to 2 senses being interpreted here. We have 2 receptors in the ear, one for sound, which is the cochlea, and one for balance, which is our vestibular apparatus, and we'll look at them separately. That little blue dot indicating the round window is going to help us absorb any excess wave so that there's no echoing in our ears. And then the eustachian tube is one that they like to ask about. That tube opens up in the pharynx, your nose-throat area, in order to equalize pressure on either side of the tympanic membrane so that it can vibrate freely. The nerves that run from the ear to the brain actually access 2 different parts. So you can see the nerves running from the cochlea. They will actually take information to the cerebrum of the brain because the cochlea is the receptor for sound, or the receptor organ for sound. And then the other nerves running from the vestibular apparatus, which relay information about balance, will actually take those impulses to the cerebellum. So when you're doing the ear, please make sure you know 2 different parts of the brain are involved in the impulses that the ear sends it.
Right, so starting with the process of hearing, if we quickly go back to our previous slide, if you look closely at the cochlea, it kind of looks like a shell that you would pick up at the ocean. And when you put that shell to your ear, you can actually hear sound in the shell itself. So I make that association that the cochlea is C-shaped, it's like a shell, shaped like a shell, and it's got to do with hearing. So if I put that shell to my ear, I can actually hear something. Right, so the steps, as Marichen has already explained to us, we're going to start with the sound waves coming in, and it's important that you use the correct terminology. So sound waves are entering through the tympanic, the ear canal, the auditory canal, and then it reaches our tympanic membrane or our eardrum. This will cause that membrane to vibrate, which will have a knock-on effect, causing the ossicles to vibrate. This will then, in result, have the stirrup hitting against the oval window. and that will bring about movement of the fluid inside the cochlea. So we refer to this fluid as endolymph, and the waves that are formed are called pressure waves. So the pressure waves move throughout the cochlea, and this will stimulate a receptor, which will create an impulse, which will then be sent through our auditory nerve to the cerebrum for interpretation.
Now, just if you quickly go back to that slide, Yolandi, that term organ of Corti, that refers to the receptor cell name for hearing. And what we're going to look at now are the two other cell receptors that are responsible for balance maintenance. So there are quite a lot of new terms and many receptors in the ear, and it's important that you distinguish between them. The cochlea, which looks like a shell, has to do with hearing. The receptor cells that are responsible for picking up those stimuli are called the organ of Corti. And now when we get to the vestibular apparatus, we're going to look at the two other receptor cells, the maculae and the cristae. Okay. So the balance is actually maintained by the cerebellum determining, what muscles need to contract and relax. So there are two sections to the vestibular apparatus that helps to control this particular function. And I've actually brought along a inner ear. Now, it's probably a little small for you, but basically there are two components. So there are these little chambers at the top that look like little loops called semicircular canals. And then at the bottom, we have a slightly swollen area that have two areas called the saculae and utriculae. And together, these will help to determine where your head is in terms of gravity. Are you looking up or are you looking down? And then the canals will help you determine in what direction are you moving. So you can move up and down, you can move left and right, and you can also move forwards and backwards. So what's going to happen with that is that the semicircular canals are actually located perpendicularly. So one of them is located in such a direction, Yolandi, maybe you can show it there on the zoomed in version, that you can see that they're perpendicular. So one of them will register movement forwards and backwards, one of them up and down, and one of them left and right. So it's an absolutely magical structure that helps you not fall on your face every time you move around outside, okay? So basically, the two parts that are important here are the receptor cells, the cristae and the maculae. And what you need to remember is which one does what. Okay, so on the slide, it says that balance is the process in which receptors in your inner ear will detect the change in the position of your head with response to gravity. So that's one receptor doing that function. And also the change in the speed and direction of your body. That's the other receptor. Now, the first receptor, the cristae, which you can see are located on the slide there in the ampulla, they are responsible for detecting a change in speed and direction. So that's the semicircular canals. Those are the three canals that can detect are you moving left, right, up, down, forwards, or backwards. So the cristae are found in the canals of the semicircular canal, so the CCC, and she's spinning in a circle, so that's going to pick up on change in speed and direction. Then the other receptors are the maculae, which are located in the saculae and utriculae, big words. And what they do is they pick up on changes in your head's position with regards to gravity. So on the picture you can see there that the maculae, when the man bends his head forwards, the maculae there are stimulated because of gravity pulling on those little hair cells. And that creates the impulse that is then sent to the cerebellum. So very important for both of these receptors. You must know their names. You must know where they're located in the vestibular apparatus. The detail of the names are not important, but you must know which one performs which function. Maculae are responding to gravity. Cristae are responding to speed and direction. Okay. So let's look at an example of a question.
Right, so as we've already gone through the different components, we're going to breeze through this. So 9.1, identify parts A to D. So A would be our pinna, which catches the sound waves. B will be our ear canal or auditory canal, which will direct it towards our middle ear. C will be the ossicles, and D will be our semicircular canals. Okay, right. Next question they want to know, write down the letter only of the part which conducts sound waves towards our middle ear. So that's going to be B. absorbs pressure waves from the inner ear, that will be G, ensures equal pressure on either side of the tympanic membrane, remember that's your Eustachian tube or Eustachian tube, that's F, then transmits vibrations to the inner ear, that will be C, and then lastly contains the organ of Corti, as we said that's the hearing receptor, which will be located in structure E. Right, they want to know what happens when part E is blocked. So we found part B, the Eustachian tube, and this has now been blocked up. Let's say you have the flu and there's a bit of mucus build up. What will be the consequence of it? So remember that its job is to equalize pressure so that the tympanic membrane can vibrate freely. So if we now block this area up, the air pressure cannot acclimatize to what the environment is surrounding us. So we could end up with a pressure buildup inside your ear, which makes this tympanic membrane less movable. So if this cannot vibrate like it should, it will result in a hampered vibration, which means that we will have less vibration of the ossicles, which means that the pressure waves would not be as intense, which means the organ of Corti is not going to be stimulated as much, and our hearing will be negatively affected. So when we say negatively affected, we're not going to be deaf, we are just going to end up with partial hearing loss. So if we go through it, ossicles will not be able to vibrate freely, the vibrations would not be able to be easily transmitted to the inner ear, causing partial deafness, or you could have said cannot equalize pressure on the other side of the tympanic membrane, which leads to pain because now that membrane is distended, and that won't be pleasant at all. If the pressure is so much, it can result in that eardrum actually bursting, which obviously will also be quite painful. And then other consequences could be that the vibrations are not transmitted yet again, and there will be partial deafness associated with it. Okay, question 10.2, what happens if part A has a hole inside it? So now when we have those sound waves that are meant to hit that thin membrane and cause it to vibrate, it's literally just moving through that membrane, which wouldn't be ideal because we rely on that membrane to catch the sound waves so that it can push onto the ossicles, so that the vibrations can be set in motion. So now we're going to have a case where the tympanic membrane would not be able to vibrate, so the sound waves to the middle ear obviously will not be passed as it should, resulting in either deafness or that the hearing will be impeded, which means that the hearing will be partially lost. Okay, question 10.3, how does part A and C work together to amplify sound? So part A is the tympanic membrane, part C is now our oval window, and what we find is that there's a size difference between these two. So if we have something that moves from a large surface to a small surface, we actually have the process of amplification that takes place. I like to think about it, imagine you have a small nail and a big hammer. If you have a large hammer that hits onto that small nail, you actually have much better job of getting the nail into the wood than if you had a small hammer and a large nail. So the same thing is happening here. We actually have that large surface area, which is now going to cause a much larger vibration, and then as a result of this small window, the impact is going to be so much more. So the tympanic membrane has a large surface area than what the oval window has.
So question 11 shows you a kind of a zoomed in magnification of the middle ear, again asking you to first identify the parts, so labels are very important. A there you can see is on the first side before the ossicles, so that indicates our tympanum, tympanic membrane or eardrum. B is indicating one of the ossicles. So you don't have to know all of their individual names. You can refer to it as an ossicle. The detail here is not important. And then C indicates the oval window right after the last ossicle that will cause vibrations in the endolymph. Give the letter of the part that absorbs waves to prevent an echo. Right at the beginning, I told you that the round window is responsible for doing that, but they're asking you for the letter of the structure, not the name. So on our diagram, the oval window is indicated by D, that membrane just below, the round window is indicated by D, just below the oval window. Then they ask you to give the letter and the name of the part that may become blocked when infected, which may result in temporary hearing loss. And as Yolandi has also explained, this is part of what the eustachian tube does in her question example. So letter E, the eustachian tube. Then another question, the last one we'll be looking at, gives you a diagram of the human ear, once again asking you for labels and functions, so make sure you understand the diagrams. Now they're asking you firstly, which parts of the brain will receive impulses from part C? Now part C is that cochlea, that shell-like structure that we have cooled down. So that part relays information about sound, and that's going to be interpreted by the cerebrum. Only information from our vestibular apparatus is going to be taken to the cerebellum. Then for 5 marks, they ask you to explain how your semicircular canals help you to maintain balance. So in this instance, you're going to be talking about what the cristae do. So notice that there are a few instances or places where you could have gotten marks. First by saying that there are cristae in the semicircular canals. What do? They pick up on changes in speed and direction. They pick up on stimuli that they convert into impulses. Remember, whenever a receptor is involved, that is usually one of the marks, by indicating that the receptor will take a stimulus and convert it into an impulse. That is going to be transmitted via the auditory nerve, if you mention the name of the nerve, to the correct parts of the brain, the cerebellum, and what happens, it helps to restore balance. So you got a mark for actually saying that balance is restored. And then last but not least, describe how an increased production of mucus in the nose and throat may lead to the bursting of part E. E on the diagram there indicating the tympanic membrane. Now, in this question, the mention of the nose and the throat should highlight the fact that we are involving the eustachian tube here, because that opens in the nose-throat area. So if mucus blocks that area, like Yolandi has explained, pressure is going to start building up in the middle ear, and the tympanic membrane will not be able to vibrate freely. But besides affecting loss or hearing, it's going to also build pressure up to such an extent that possibly that tympanic membrane could burst, and this could lead to a lot of pain and partial deafness. And that's the ear and the eye for today. That sums up all the revision we've done for term 3, and we want to wish you good luck with your exams.